Welcome back, everyone. Next up is Bioxytran, Inc. They're an OTCQB company trading under the ticker symbol BIXT. The company is a clinical stage biotech with 3 revolutionary platform technologies that were quickly developed using the power of AI. They have a broad spectrum oral antiviral drug that recently completed clinical trials and achieved 100% viral reduction in 7 days, a feat only accomplished by one other drug in the past decade. They recently published research on their historic discovery in virology, which debunked the 50-year-old notion that for every virus, you need to develop a different antiviral. Their drug is a galectin inhibitor that not only neutralizes COVID-19, but also neutralizes influenza and RSV and prevents it from entering cells. They are still recruiting patients for their dose optimization study, which will give them the ability to proceed with their registrational trial in India after they finish. They have a second platform drug, an oxygenation molecule being developed to treat stroke, dementia, and sickle cell anemia. Now, due to the molecule's small size, it has the ability to carry oxygen into areas of poor circulation. The company also has a third platform technology that's capable of treating cancer metastasis. And Mike recently wrote a LinkedIn article on how galectins are the primary reason why we don't have 90+% response rates in cancer immunology treatment. Mike Sheikh, Executive Vice President, Business Development. Always nice to have you back on the conference. Now, we're going to change it up a little bit today. For the past few conferences, we usually do an interview style, but I think you've revamped your investor deck and you want to do the full presentation this time. So we're happy to have you back.
Yeah, I'm raring to go, Anna. I'm raring to go.
The floor is yours.
Okay. Well, I want to thank all the investors for being here today. What we have is a new slide deck, a revamped slide deck. So I'm anxious to show that to investors. There's our forward-looking statements. Read them at your leisure. Let's start off with the investment thesis, okay? Bioxytran has game-changing platform technologies, not one, multiple. And our clinical trial results pretty much validated the science, which means we have a high probability of success in any clinical trials that we do. And we have the IP, we got the proprietary science, we got all this AI technology that helps us craft new drugs. And why is that? Make this world a better place. I mean, that's kind of what we're about. And when investors look at the market cap, they're going to see an extremely compressed valuation, given that we're essentially a phase 3 ready asset. So it's really at an ideal buying level. We've got a solid management team. We got a solid strategy to make shareholders money. And there's also opportunity because we have a thin float, we have a loyal shareholder base, and that in OTC leaves the possibility for explosive upside moves. And the other thing I want to point out is we are in active licensing discussions. And I'll get into that a little bit more. Let's start off with our revamped mission statement. Bioxytran is a clinical stage pharmaceutical company focused on neutralizing chronic inflammatory diseases using carbohydrate design. The drugs are expected to have major impact on the fields of glycovirology, oxygenation, and immunology. But now you see right in the center, I want to focus people's attention. We want to take inflammation out of your lives permanently. That's our true mission. Now, how are we going to do this? Well, we have drugs made of complex carbohydrates. They're going to treat viral infections. We're going to do oxygen transport for hypoxia, and then cancer and fibrosis. Now, our carbohydrate drugs recognize this specific receptor called the galectin, and we're going to get into more of that a little bit later, but just remember that word galectin. Now, what do? We make antiviral drugs, we have a miniaturized blood carrier, and we have a cancer metastasis / fibrosis type drug. And what are these drugs? Well, prolactin M, that's an oral galectin inhibitor. And you can see a little virus, it treats viruses. We got prolactin I, that's an intravenous galectin inhibitor. And that's really more designed to go after tumors and fibrosis. And then, of course, we got BIXT25, which you can see there, the little vein and the oxygen going there. That's our oxygen transport molecule. Now, when it comes to biotech, like one of the first things that you want to do is you want to check out the leadership and the management team. That's critically important. And what we have here is Dr. David Platt. He's the CEO of our company. And he's the guy that coined the name galectin. So remember I mentioned the word galectin? This whole field of research around galectins started with him in 1992 when he coined the name galectin, and then shortly thereafter published one of the first articles in that field of research. So here we are. He has 40 years of experience researching galectins and carbohydrate binding proteins. There's over 4,000 journal articles talking just about these galectins and in cancer. And then he's got textbooks, he's got journal articles, patents, clinical trials, 5 public companies. When you want to talk about a pedigree in galectin science, Dr. Platt has got it. And we're very pleased to have him on our team. Now, this slide is going to be very interesting for shareholders because this is how we are going to make money for you. Our job is to develop these drugs to the point where we can get licensing deals. We have done our job. Now we're in the thick of it trying to get licensing deals from big pharma. So now let me explain this. You notice the big iceberg, and below the iceberg you have all these names. Those are all big pharma names. Now on these big pharma names, you're going to notice oncology, neuroscience. I mean, there's a whole myriad of therapeutic areas that each big pharma has a specialty in. And then you'll notice at the top, those are the specialties that our galectin science can treat. So you kind of see the overlap. There's really not one big pharma company that we can't make a case for our technology. So when it comes to big pharma or little pharma for that, man, I got to tell you, there's very few people who we shouldn't be or can't talk to. And that's pretty exciting, especially I'll let you guys in on a little secret. So breaking news, we are going to go to JP Morgan, and one of my licensing guys is starting to book me meetings at the conference. And he said space is filling up fast. So we'll see what happens. Now let's move on to glycovirology. And our key molecule there is prolactin M. Remember I said I was going to explain galectins. So galectins are protein receptors that recognize sugar molecules. So again, galectins are protein molecules that recognize sugar molecules. Now, that's great. What do? Right? And then you see the little graphic. There's these little receptors that come up, and there's these little sugar spikes on the end of those receptors. That's where the galectins attach. And they're right there in this little graphic. They're responsible for signaling, but they're also responsible for adhesion. So they're adhesion molecules, and they can bring cells together. They can bring receptors together to signal something. And what else can they bring together? And we'll get into this later. They can bring viruses together. They can help viruses stick to cells. Now, the inspiration for virology came from all these journal articles down at the bottom there. You'll see all the footnotes and stuff like that. I'm not going to read this to you, but the bottom line is we we read some literature, and we came up with an entry inhibitor really fast. Early back in I think it was February of 2020 is when we started this journey, and we figured out the properties for an adhesion molecule that plays into things. Now, let's talk about the historic discovery that happened in virology that really I don't think anyone's paying attention to. But I want to stress to investors just how significant this discovery is. For 50 years, there's this notion that one drug can treat one virus. And this has withstood the test of time for 50 years until our drug, Prolactin M, comes around and completely debunks this idea. Prolactin M has broad spectrum activity in COVID-19, RSV, and influenza. And to make things even better for investors, we have a patent for 60 plus viruses. So we could try our molecule, Prolactin M. We could try it on 60 molecules if we want to, and we're covered by that patent. So that's pretty darn exciting. Now you're going to ask, okay, so what exactly is this discovery? Well, the discovery is this concept of viral adhesion. We need viral adhesion to the cellular membrane, and that's the first step in the process of viral entry. And galectins, they facilitate this process. So both the viruses and cells, they're basically covered in these slippery membranes, whether it's on the spike protein or whether it's on the actual cell itself. And galectins, they kind of act as this viral glue between the two so that they can tether the virus to the cell. And then that's what helps begin the process of viral entry. Now, existing antivirals work on a one virus. They work on one virus or maybe some mutations of the virus, but they're not really broad spectrum. They might say they are, but they're not. And they work on a different mechanism of action. They disrupt the life cycle of virus. They only work once the virus has infected the cell. We're doing something completely different. Actually, I don't know many that are doing what we're doing, but we're trying to block it outside the cell, never let the cell get infected to begin with. And look at that next point. There are no broad spectrum antivirals that currently exist. And what are we going to do? With this technology, we have the potential to disrupt a $55+ billion global antiviral market. So what investors need to do is ask, wait, with all this technology, does an $8 million market cap make sense? I think you know what I think about that. So now let's look at our platform itself. It's versatile. We don't have to worry about mutations. It's tested. We've had a couple of clinical trials. No safety issues, non-toxic. No expected limitation. What does that mean? We can give it to young people, old people, pregnant people. It doesn't matter. And then it's efficient. What does that mean? That means we don't spread it. It eliminates the contagion and the spreading from one person to the other. So we would consider ourselves the first line of defense against all mutation of coronaviruses. Now, what does that mean? That means if you get sick, you take these things, you feel better, and you're over it. That's what this means. So what clinical trial stage are we? Notice there, we're phase 1B in the FDA and phase 2-3 for India. Now we're going to let me talk about the summary of our clinical trial results. But before I get into that, let's just give you some breadth idea of how big this is. Okay, so there are 10,000 clinical trials that were done in approximately the last 10 years. Who ranks at the top of this list? We do. We are number one. We have the best, because you can't get better than 100%, right? We have the best phase 2 clinical trial efficacy data readout in the past decade. So all those other biotechs don't have as good of efficacy as we do. They might have equal, but they don't have better. All right. So what does that mean for an investor? As an investor, what that means is you have that our clinical trials have an extremely low risk of failure. We had a 100% responders rate and a p-value of 0.001. That is literally unheard of in biotech. So we're very special, and I want investors to really understand that. And I want you to do your due diligence and dig deep and see what we have. Now, what are the results that I'm going to summarize? We eliminated the virus and symptoms within 5 days. I'm going to show you that. We reduced our infectivity, which means we're not giving it to other people. We quieted the cytokine storm. What does that mean? You don't have to go to the hospital. And we have a robust antibody response. We call it post-infection immunization. What does that mean? That means if you get, you're not likely to get sick again. Now let's look at the data. The chart on the left comes from our peer-reviewed clinical trial review or journal article. And you'll see there's a tight grouping between the treated and the placebo arm. And you'll see the red is the placebo, the green is the treated arm, and the goal, and this represents viral load, they call it CT values, but you basically want, you're all at the same starting point, and you want the lines to dive through the top of that dotted blue line. And you'll see what happens is that the green viral loads, they're going up, but they're actually going down. That's what CT value means. Dive right through there at day 3 with most of them. And you see day 3, look at this, 88% were PCR negative. Day 7, 100% were PCR negative. Now that's versus only 6% in the control. There's like this one guy that you can see just sticking above the line. Now let's get into what our phase 3 is going to look like. It's hard for me to say phase 3 an $8 million market cap in the same breath when other phase 3 companies have like $500 million market caps and higher. It boggles my mind how disconnected from reality investors are. But anyhow, we were going to go after 408 participants, double-blind, randomized, placebo-controlled study. We're looking at the change in seropositivity at day 5. That just basically means, hey, you're PCR negative or not. And we're broad inclusion criteria. We're going after standard risk patients. Well, I'll talk about that on my summary and why that's important. What else did this study prove? We don't have viral rebounds. See, Paxlovid, there's a graph of Paxlovid there, and the chart shows how the virus goes down, and then right at day 10, bam, it comes right back up. You can kind of see that on the chart where the dotted break of the dotted line is. We had no viral rebounds. And what else did we not have? We had an absence of symptoms. Look, both the placebo group is in orange, and you'll see the orange lines represent symptoms, and then and day 7, there's still kind of a bunch of symptoms. They went down a little bit, but there's still a bunch of symptoms. But there's almost virtually no symptoms in the treated group because you feel better very fast. Now let's compare to Paxlovid. If you look at these charts, number one, there is no fair comparison. There's no head-to-head. We didn't do a trial with Paxlovid. This is just real-world data versus our clinical trial data. All right, but here's what I really want to say. Look at where we cross the green line on our trial. It's a day 1.5. So 30% of the people are better or are basically taken care of, PCR negative, in about a day and a half, according to this chart, versus how much? 30% line? Yeah, it's going to take 20 days with Paxlovid. So do you want to be sick for 2 days or 20 days? I think the answer is simple. Now, here's an update on our dose optimization trial. I know there's some investors out there that are kind of frustrated, but we've had slow recruitment, and only 12 out of the 40 patients in this clinical trial have been dosed. The issue has been we're trying to get people with no underlying medical condition, and in India, that is harder than what you think. The only people going to the hospitals are really the people with underlying medical conditions. But investors need to know, look, this trial is fully paid for. There's no money back guarantee on this thing. And the recruiting sites are still open, so who knows? But our endpoint is basically dosing data so we can get to a phase 3. The final thing on the study that I want to talk about is this idea of post-infection immunization. The idea is because we clear the virus so readily with our antiviral or entry inhibitor, it really creates a strong, robust adaptive immune response, which means if you were to get sick again, you have a lot of good protection there. And then I wanted to stress the broad spectrum antiviral activity again. And let me see, I'm not sure if it's showing here, but there's all the, a lot of people say, oh, yeah, I've heard of a broad spectrum antiviral. No, you haven't. You haven't heard of it. You've heard of broad spectrum antibiotics, and that's what I wanted to show you. There's broad spectrum antibiotics, but there are zero broad spectrum antivirals. I mean, the ones that claim they're not broad spectrum. So you see this little marketplace here? That is ours for the taking. We're the only guys in that space, that $55 billion market space. That's what investors should get excited about. Now, how does it work? We neutralize the spike protein. You see, antibodies normally attach to the tip of the spike protein, and that puts like a coating on it, prevents them from entering, and targets them or makes them noticeable to the immune system. But what we do is we go on the side, and you'll see this sugar-coated picture there of a spike protein. So there's a spike protein without on the left without sugar, and there it is on the right. Notice the blue areas are the only areas where anything can attach to, right? Otherwise, it's slippery. And where the red circle is, that's our binding point. That's the galectin fold. So what we do is we bind up the virus, like in this graphic, and then it kind of acts like a Swiffer. Our molecule's big. It's like this big Swiffer, and you have all these dirt particles represent viruses, and then so it's not like one molecule, like one antibody to one spike protein. It's like one big molecule for hundreds, thousands of virion, and then that one molecule is going to get disposed of by the liver or filtered out by the liver and excreted. Now we have other indications. We've done a case study on shingles, looked that up, viral conjunctivitis. Another thing that we have is long COVID. We got a bunch of theories on long COVID. And guess what? Galectin-3 is implicated in pretty much all these theories of long COVID. So now I'm running low on time, but that takes me to my next topic here, which is our oxygenation platform. And the key there is BIXT25. Stroke is a big problem. I think we all know that. It's big. Why? Because you got your caregivers. Caregivers are spending, it's in the billions, okay? And it's in the hundreds of billions. I would say trillion is probably a better number. I think these estimates are low. But be that as it may, what's driving this issue? What's driving it is the time that it takes to get treated. So follow along right below the graphic there. So there's the onset, you could see that you have a stroke, and then the ambulance comes, and then you go for imaging, and then you get the procedure done, and that's kind of how it's done. And the reason they do this, it takes 2.5 hours, but your brain all this time, it's holding its breath. There's no oxygen going through. We need some sort of bridge. And wouldn't the perfect time be for a bridge? As soon as the paramedic sees, oh, wow, you're having a stroke, and they're going to put an IV in to treat you for shock, right? Why not give the oxygen bridge? And here's the oxygen bridge. It's BIXT 25, and you can see all the blue there, and you can see the clot. This is a cross-section of a clotted artery. And what happens? The blue oxygenated molecules are flowing right through. They're flowing through with the plasma, no problem. They're not getting clotted up. And just keep in mind, this is like, the size here is not right. It's 5,000 times smaller than that blood cell. So it's like a little speckle. So it's just right in with your blood plasma. Now, how do we make it? We take blood, that's our main product, and we break it down till we get the heme. And heme is what carries the oxygen molecules. And we stabilize it with what? Our carbohydrates, right? So our carbohydrate drug stabilizes the heme, and it's delivered as an IV solution. There's no way we're ever going to make this into a pill or anything. The immune system doesn't see it, so you don't have any of that blood typing issue type stuff when you take this. And it's shelf stable for a while. And where are we? Well, we've done proof of concept in animals. We've done it on albino mice. We've done it on rabbits, and we're now going to do larger animals, pugs. And then what's going to happen is we're going to submit it for an IND. And then this graphic here shows like an earlier proof of concept, where what we're doing is we're kind of blocking off the main artery, and oxygenation is still going through. That's what that picture shows. So it shows proof of concept in stroke. And then prolactin I. Hopefully I'll be able to finish this quickly. This is cancer. This is all about cancer. The ultimate cancer target is going to work on multiple indications of cancer, multiple disease stages, and relevant for all combination therapies. And galectins are implicated in all this. Just read up on that. The next slide here talks about the galectin signature in cancer for all different types of cancer. Brain cancer, you can see melanoma, esophageal cancer, breast cancer, kidney, all that. So 30 years have gone by, 4,029 journal articles, all on cancer. The biomarkers all say cancer, yet there's no galectin inhibitors yet. So bottom line is galectins are essential to tumor survival. I mean, galectins are responsible for all these functions in cancer, which keeps cancer happy. So if we take away the galectin from the cancer's perspective, that's like leaving this. Look at this kid. He's got no food, no water, no clothing, no shelter, and he's out in the middle of the desert. That's what you're doing to the tumor when you take away their galectins. That's why we want to block them. And then galectins impair the immune system. And this graphic, man, this shows it to you. I mean, you can read up on all the mechanisms there, but this is what's called a Kaplan-Meier plot. And look what happens. The green is the person with low galectin expression. The other one, the downward sloping one, with 5% probability of survival, 5% survivability, right? Has high galectin expression. Who do you want to be? You want to be the guy up here on the green slope, or you want to be the guy down there? That's why you want to block galectins. So what happens if you block galectins? I know the answer to that. It's right here. Clinical trial results were done way back in 2018. The whole world's been sitting on the best clinical trial data in cancer for over 5 years. Belapectin had 100% objective response in I think it was the second cohort, and 1 in 5 had a complete response. Pretty darn amazing data. Just look at the before and after picture. This guy is riddled with cancer, and then 180 days later does a PET scan, he's clean. Just crazy that we're sitting on this technology. And you should listen to my last cancer presentation. I talk all about it. Now let's get into our business development strategy, because I'm really running low on time here. This is our pipeline. You'll notice Prolectin M, that's at the top of our list. That's our oral antiviral, chewable tablet, and we're going after viruses. And the next one is Prolectin I. That one's ready to go. We need funding for it. Actually, we need funding for both, but the idea is I just want you to know that both drugs are made and ready to go. And then BXT25, yes, we've made the drug and we're doing animal testing with it. It's not quite ready yet for human testing, but it's definitely moving along. And then what's our business strategy? So investors, this is what you got to pay attention to. We are all about licensing. That's how we're going to make you money, is by licensing this product. And the first thing that we could do is we could do a licensing strategy for India. And if we do, we anticipate we'd get like around $30 million and around a 5% royalty. And that's coming from some industry experts that have kind of given us that information. But the other thing that we can do is we can partner with small or big pharma or government. That's something new that I added to the list. Anyone notice that? Hint, hint, stop, stop. Now, I have two licensing people that I'm working with. One's focused on Japan and one's focused on the U.S. market. So I'm getting those I'm planning on making those type of connections and meetings at the JP Morgan conference. So that's something to listen to, and there could be some catalysts. But the other catalyst, don't forget what I mentioned last time, is Galectin Therapeutics is coming out with their readout supposedly at the end of the year. I think that's going to raise the awareness level of this galectin science big time. And we could see a real value inflection point. So don't discount the power of that either. And then here, I'm talking about this, I didn't move ahead on the slide, but this is the competitive landscape. You have Galectin Therapeutics, which I just talked about. You have Galecto Biotech, they make small molecules. And then you have True Binding. And then look at this, they're in all these diseases that are kind of long and chronic. I mean, the cirrhosis trials are long, Alzheimer's long, stroke long, COVID-19 5-day trial. So our strategy is about getting a viral clinical trial done, whether it's COVID-19, flu, influenza, we'll figure it out. And after that, we can do label expansion. So now we have IP, you can look at that, but now let's look at my summary slide. Okay, so, and I think I probably got a minute left. We got experienced management team. We got cost-efficient development. That means we are fiduciary of your investor money. We've de-risked the science. We're a value inflection point. Look at our stock price. It's in the gutter. It's ready to go. We have a bunch of loyal shareholders, and we're aggressively pursuing licensing. What's not to like about this? And then finally, there's our management team. And I don't think I have time for any more questions, but I'll let you be the judge of that.
Yeah, unfortunately, you don't have time, but we do have lots of questions for you, Mike. So we will send you these questions. You can answer our viewers on your own time, but always fascinating work you're doing. And I think 2025 is going to be your year. We look forward to following you along with your updates. All right, everyone, we'll be right back.